Oh, hello, sir. How may I help you?
Hello, Professor. I'm Patrick.
Okay.
And I was wondering if I could interview you today.
Of course, of course. May I know what the topic is?
Well, I'm sure you've heard about the recent alien that was discovered.
Oh, yes, yes.
And
That was captured a few months ago.
Yeah, and I was just wondering if you would like to talk about some of the similarities and differences between the alien DNA and human DNA.
Okay. Well, after taking some cells from the alien, we did observe that there was one nucleotide that was similar compared to our human DNA, which is adenine.
Right.
Another similarity we saw was that the DNA came in a double helix structure. Right. And since due to this double helix structure, we could assume that there was hydrogen bonds between the nucleotides, as you should know.
Now, what about the other three nucleotides, such as guanine, cytosine, and thymine?
See, what we found was that there were isoforms of thymine, adenine, and cytosine. But as for guanine, there was in a beta form, which is not an exact form of guanine, and it wasn't being used in the same place as well.
Okay.
All right. So with this, we could see that there's 3 base pairs instead of 2.
Right.
Which is the difference between, since we have a total of 4, but in the alien DNA we could see that there's 6 instead of 4.
Now, if some people may not be aware, human DNA runs anti-parallel.
Right.
The 2 strands run anti-parallel, which allows for the hydrogen bonding to occur.
Correct.
Now, being that there's 3 base pairs, would they run anti-parallel or parallel?
See, what we found was that this is a human DNA structure. Alright, so this is one strand, this is another strand. And 5 prime, 3 prime end, as you know,
Right.
ran anti-parallel.
Right. To allow for the bond.
Right? Okay. So, what we found in the alien DNA was that it ran parallel. So, the alien ran 3 prime, 5 prime, or vice versa.
Right. And 3 prime.
Or vice versa. Now we pondered upon this question and thought about how this was possible.
Yeah, because that would not make sense if that was...
Because we understand that adenine forms a hydrogen bond with thymine,
Right.
and then guanine forms hydrogen bonds with cytosine.
True.
See, what we discovered was that since they're isoforms of those nucleotides, they were able to form hydrogen bonds with the remaining nucleotides. For example, one of the main differences we saw in the alien DNA was that uracil was present, which we only find in human RNA.
Yeah. How would that work?
See, what's going on is that since uracil can take the place of thymine, that allows the hydrogen bonds to form when it's parallel. Same goes when the isothymine, cytosine, and adenine are added into this picture.
So would the functional groups bond better in the isoform in a parallel state?
Yes, see, in humans, the functional groups are anti-parallel. So basically, it's positive and negative.
Because they're going to attract.
And negative and positive molecules at the ends of the nucleotides, which attracts, which forms the hydrogen bonds, right? Now what we can see after adding the remaining nucleotides, which is the isoforms of them, we saw is that instead of them repelling each other, it's the opposite attractions. So we saw negative and positive, positive and negative, thus forming the hydrogen bonds.
Okay, well, thank you for your time.
No problem.
I think that about wraps it up. So
Right.
I'm going to go take this to the New York Post and see if we can get a story made out of it.
No problem.